something at the bottom.
That's like the full screen.
11.
Try F11. Or you can press like the full screen.
Okay. So this is my second high-level question. How does the environment impact DNA and gene expression? So what is a mutation? The environment can change DNA in good ways and bad ways. Mutations in DNA happen when a person's DNA gets duplicated, removed, or changed permanently. Sometimes these mutations have no effect on the body cells. Other times, they can be really harmful to them. And what is gene expression? If a gene is expressed, it can be used to make something like a protein or mRNA function according to gene expression and regulation. So, while 99.5% of the genome is the same between individuals, 0.5% of the genome makes each individual unique, and it gives them traits like eye color, hair color, like your height, and more. One type of genomic mutation in the DNA is the short nucleotide polymorphism. It's when a few nucleotides up to 5 get mutated. Scientists research how SNPs change traits in DNA. So there are many different types of mutations. Here are a few. So if this is the original sentence, red dog and fat cat and fun, R could be substituted by F, which turns to red dog and fat cat and fun, which makes no sense. And there's also deletion, where all of this is deleted, and then it turns to red dog fun, which also makes no sense. And if something is inserted, like as you can see, you are here, it turns to red dog and fur and fun. And the reason why the rest of it looks like nonsense is because whenever an insertion happens, every line gets shifted to the next codon because codons are different in terms. Also, there is a fusion of two sentences and multiple copies of the same sentence. So here's an example of a DNA mutation. As you can see here in chromosome 2, G is turned to A, which changes amino acid arginine to amino acid histidine in protein IDH1. It causes breakthrough. So you may be wondering what IDH stands for. It actually stands for this really long word called isocitrate dehydrogenase, and so people just call it IDH. So brain cancers. Gliomas are a common type of brain tumor. About 30% of gliomas are gliomas. Brain tumors are gliomas, and 80% of these tumors are cancerous. The IDH1 and IDH2 genes carry the instructions to make two enzymes or proteins, very important for brain function. These enzymes are involved in regulating metabolism, which are reactions to make energy needed in the cell. But mutations can alter their normal function and drive them to become cancerous and multiply too fast and too much. Over 80% of lower-grade gliomas contain IDH mutations. Drugs are currently in development to alter mutated proteins. So, a mutation in this gene causes sickle cell anemia. So sickle cell anemia is a disease caused by a mutation in the hemoglobin gene. As you can see here, T is changed and A is changed. And T to A mutation changes amino acid E to V, and the T to C mutation changes amino acid E to A, and the T to G mutation causes amino acid E to turn to G. And if a person gets sickle cell disease, their red blood cells change from a normal shape, which is round like a button, to an abnormal shape, which resembles a sickle, like the head of a sickle, which is how the disease gets its name. You can also compare it to like a crescent moon. This can block blood flow, leading to a lot of pain, infections, and organ damage. So here Here are some facts about sickle cell anemia. Sickle cell disease affects more than 100,000 people in the United States and 8 million people worldwide. So in December 2023, the U.S. Food and Drug Administration approved 2 new gene therapies that are life-changing for individuals with sickle cell disease. One treatment adds a modified gene into the body, and another makes a change to a gene that is already in it. So, leukemia is caused by a fusion of 2 genes, BCR and ABL. So it's caused by when a piece from chromosome 9 and a piece from chromosome 22 break and switch places. And a normal chromosome is made when pieces from chromosome 9 and 22 break off and trade places. The ABL1 genes from chromosome 9 breaks off and trade places with the BCR gene in chromosome 22 to form the BCR-ABL1 gene, fusion gene. And the chromosome 22 with the fusion gene on it is also known as Philadelphia chromosome. The BCR-ABL fusion gene is present in around 95% of leukemias. BCR-ABL1 genetic laboratory test uses some sample of blood or bone marrow to look for the abnormal fused gene. Drugs are available to target the BCR-ABL fused protein and treat leukemia patients. So, environmental exposures can cause DNA damage, like UV rays from the sun, smoke from burning, some foods processed at a high temperature, drugs, medicines, and chemicals. There are a few types of DNA damage, such as interstrand crossings, where a nucleotide attaches to the nucleotide next to it, not across from it, and bulky adduct, when new base pairs are added. Interstrand crossings, when a nucleotide attaches to the nucleotide diagonal from it, and double strand break, where both strands of DNA are broken in one place, and single strand break when DNA is broken in one strand. So, how does the environment impact DNA? Things like exposure to UV light, radiation, and chemicals in the food we eat can change your DNA by a mutation or by epigenetics, which is a chemical modification of DNA. So, we've talked about translation and transcription and also mutations, but now we're going to talk about how AI can help find the meaning of a DNA sequence. So this is how can AI help in decoding genome sequences. So AI can help find the meaning of a DNA sequence by looking at a codon chart and then determining what patterns and what amino acids are being made into a protein. It can also help by finding patterns in DNA, such as how the start of gene is always ATG, and TAA, TAG, and TGE are always the end of the gene. So artificial intelligence, AI algorithms, for example, DNA Expecto and AlphaSense, can analyze DNA or amino acid sequences and identify mutations that change the function or the expression of mutated DNA or protein, just like in an English sentence. So this is how AI can help doctors. If a person got the disease that had something to do with their genes, or if their genes had a mutation, AI could look at it and find out where the mutation was and what went wrong. Since doctors can't read through all the DNA, AI does it super fast and then finds out where the mutation was in the most important parts and then shows it to the doctors, and then the doctors can help care. So this is how AI can help doctors. So a patient is sick and they visit the doctor. Their blood is drawn and it is taken to the laboratory for a few tests, including which may include the genome test. Since doctors can read the whole genome sequence, about 3 billion nucleotides, AI has to do it. So AI reads through it and then figures out the mutations and sends a report back to the doctor who treats the patient. So before I show you the tactile, I interviewed two people, Mr. Max Chow and Mr. Matthew Nicholson. So here are the links. So my name is Lee and